Hello friends, today we are going to discuss one about the important topic in genetics about sex-linked inheritance. Now, sex-linked inheritance. We all knew that genes controls the inheritance of traits. Does this mean both the autosomes and allosomes have the same pattern of inheritance? Not exactly. Allosomes means sex chromosomes. Here in case of autosomes, the probability of inheritance in both the sexes is same. But in case of allosomes is not. Depending on that means the allosomes determines the inheritance pattern. That means depending on the kind of the sex, whether it is male or female, the inheritance pattern will be decided. And such pattern of inheritance, that means the traits controlled by the genes located on the X chromosomes or Y chromosomes. That means the one of the sex chromosomes are called sex-linked inheritance. With respect to the beginning, Thomas Hunt Morgan is the first pioneering geneticist who has worked out and consolidated and give a concrete evidence that the sex-linked inheritance pattern is on board. What exactly I meant to say is, he has performed his famous Drosophila melanogaster eye color experiments, and which concludes that there are specific genes associated with specific chromosomes. To be specifically, sex-linked genes determines certain patterns. Okay, what exactly has done? Thomas Hunt Morgan, popular known as father of modern genetics, just like the Greek Johann Mendel is the father of genetics and he is the father of modern genetics. Say he has done experiments on Drosophila melanogaster, what we call fruit fly. Okay, say the two kinds of varieties he has picked up. Say a red-eyed normal female. Here, red-eyed is normal, wild type, whereas white-eyed is a mutant. Okay, a cross between red-eyed female, that means normal female, with a white-eyed male, that means mutant. Red-eyed, that means normal, and white-eyed, mutant variety, male. Okay, we knew that gametes will come, it's pair generation. Gametes will come, two kinds of gametes will come. Then the crossing between these two leads to two different kinds of offspring. We knew that. Here the female is a carrier. That means heterozygous. Whereas males are normal. Males are normal. Then this is up to F1 generation. Okay. Then he has crossed again a carrier female with a normal male. This time here, in case of first crossing, the male is affected. But in case of next crossing, the male is normal. But the female is carrier F1. Okay. The carrier you can draw here. The carrier XR and X small r versus normal male, normal and carrier. We knew that checkerboard we can draw Punnett square. Okay, the two kinds of gametes will come here. So female and male, XR, and the carrier and normal male. The first case, that means the first daughter is normal, and the first son, that means not exactly first, just for convenience. Okay, one of the son is not normal and one of the daughter is carrier, but for now is normal. That means red eyed, but another one, the son, is white-eyed. That means 50% of male population are affected in is called F2. So the point is clear. Even then when the Morgan crossed a normal red-eyed, a wild female with that of a white-eyed, a mutant male, he got a carrier female and a normal male. But when he crossed a carrier female of F1 generation with a normal male, he got 50% of the male affected. This is called XY, that means sex-linked inheritance, is specifically called X-linked inheritance. X-linked means, here I told you already, genes located only on the X chromosomes and whose alleles are absent on the Y chromosome are X-linked genes. Similarly, Y-linked genes, genes which are present on the Y chromosomes whose alleles are absent on the X chromosome are Y-linked genes. We will discuss about later about XY-linked gene species. Okay, for now just listen. Here the character, that means the affected male, what we call, say, a father. The character of the same, that means the inherited mutant has been inherited to that of the grandson through a carrier female. This is called skip generation of inheritance or criss-cross inheritance. Criss-cross inheritance or skip generation inheritance. Criss-cross means, say, here he is a father, that means he is a grandfather and he is a grandson. Criss-cross is like criss-cross, the same here. That means here the grandfather to grandson through carrier F1. Criss-cross. Skip generation means here the F1 generation male has been saved. F1 generation male has been saved, but F2 generation grandson has been affected, because this grandson got inherited the affected gene, that means the affected mutant allele through F1 daughter, which actually belongs to his grandfather. So inheritance of a mutant allele from grandfather to grandson through the carrier of the first generation daughter is a skip generation or criss-cross inheritance. And Morgan for the first time clear-cutly gave a consolidated proof that specific genes are associated with specific chromosomes. That is why he aptly considered as a father of modern genetics, because he is the chief architect. Okay, so it's about the first cross, and he also done the reciprocal cross. The reciprocal cross means same but different parents. Reciprocal cross, why he has done a reciprocal cross? Reciprocal cross is to understand the parental sex role in the inheritance. Say in case of reciprocal, the same concept but different. It's a red-eyed female versus the normal male. Red-eyed versus normal. Sorry, it's white-eyed. Sorry, this is white-eyed. Okay, this is normal. Okay, small r denotes white-eyed, capital R denotes normal. With this is a simple logic to understand. Normal. Okay, normal red-eyed. Then we got, as usually, we got one kind of gamete, and here we got two kinds of gametes, and the crossing leads to again the carrier daughter, because capital R is normal, small r is affected, but the son are affected here. Then this is called F1. Then F2. It's the same concept, but it's a bit different. Carrier daughter with that of a white-eyed male. First, a carrier daughter a normal son, then an affected daughter, and an affected son. That means 50% of children are affected. Here, when Morgan done the reciprocal cross, that means between white-eyed female with normal male, in the first generation itself, the males get affected. Males got affected. Whereas in case of normal crossing, the first generation males didn't get affected. That is why I told you, reciprocal cross to understand the role of parental sex chromosomes in the inheritance. Here the first generation male itself he got affected because he got the one of the the mutant allele from the mother has been inherited to him and he got affected. In case of F2 generation, in the F2 generation, 50% children got affected. So that means the pattern is completely different. But in case of autosome, it is not like that. The pattern of reciprocal cross also same just like the normal cross. That means in case of autosomes, that's important point. Same reciprocal cross results. Same reciprocal cross results. But allosomes will have different reciprocal cross results. Different reciprocal cross results. This is very important. I told you now. There is a difference of inheritance pattern depending on the kind of chromosomes. Autosomes and allosomes. Autosomes simply both sexes have the same probability of inheritance, but here the difference is depending on the sex chromosome, depending on the kind of allosome. That what actually the role of sex-linked inheritance. Okay. Then it's about the what exactly the Thomas Hunt Morgan has done, and based on it we can discuss the remaining issues. We have discussed already the most important point. Sex-linked genes. Sex-linked genes. Sex-linked genes. What are sex-linked genes? The genes which are located on one of the sex chromosomes, say either X or Y or X-linked genes. We should not confuse between sex-linked genes with XY-linked genes. I will tell you how. The sex-linked genes means genes which are present on either X or Y. And here again there are different patterns here. In males we knew that males, one is X, another is Y. In females, both are X. That means males are hemizygous to both X chromosomes and Y chromosomes. That means to inherit a trait, one copy is enough. With respect to X also and Y also. But in case of female, depends on the kind of mutation. For example, if it is recessive, 2 copies required. If it is dominant, 1 copy is required. Because they are either homozygous, that means homozygous, or heterozygous with respect to female, but males are hemizygous because 1 copy is enough with respect to both X and Y. So depending on the kind, there are different patterns. That means so pattern varies depending on the sex, depending on the, that means because depending on the allosome simply. Say the sex-linked genes. Say so here X-linked genes and Y-linked genes we have that. With respect to X, that means X-linked genes means the genes which are present only on the X chromosomes whose alleles are absent on the Y chromosomes are X-linked genes, X-linked genes. Whereas Y-linked genes, the same concept, genes which are present only on the Y chromosomes whose alleles are absent on the X chromosomes. No X chromosome and no Y chromosome here. And with respect to X-linked genes, for male, since they are hemizygous, 1 copy is enough. And for females, depends, both copies, because sometimes it depends if it is recessive, both, or if it is dominant, 1 copy. Because the homo or hetero conditions are possible in females because 2 copies of X chromosomes are present. But in case of males, just both, with respect to both allosomes, 1 copy is enough. Why? We knew that only male get inherited from his father, and there is no role for females. Okay. Then we have discussed another concept, what we call XY-linked genes. We should not get confused between sex-linked and XY-linked. There is a bit difference. Sex-linked genes, this is very important point, do not undergo pairing or crossing over. This is very important. Sex-linked gene, that means either X or Y, they do not undergo crossing or pairing. Crossing over, for example, doing packaging stage of prophase 1 of meiosis 1, they do not undergo. That is why they are specific. They have a specific kind of inheritance. But X5, the autosomes undergo pairing or crossing over. Here, X5 linked genes, actually, the homologous regions of X and Y chromosomes, they undergo pairing or crossing over, just like autosomes. That is why they are considered as incomplete sex-linked genes. Incomplete sex-linked genes. This is very important. In examination, they may ask you what is the distinction between sex-linked and XY-linked genes. XY-linked genes, just like autosome, they also undergo. That means the homologous regions of XY chromosomes, they undergo pairing or crossing over. That is why they are considered as incomplete sex-linked genes. Here, they are considered as pseudo-autosomal regions, the part which involve in crossing over. So they are different. But here, sex-linked genes are complete. They do not undergo pairing or crossing over. That is the essential difference. Okay, these are sex-linked genes, and these are about XY-linked genes. And this, with respect to males are hemizygous for both X and Y. In case of females, both homo or heterozygous, depending on the kind of disorder. 2 copies are required to get expressed in case of recessive, that means XX. 1 copy is required in case of the dominant disorder. Okay, so both sex-linked and XY-linked genes. This is essential for further deliberations. Okay. Then, as I said, the most important topic, sex-linked inheritance. So, if we understand the Thomas Hunt Morgan experiment, it is easy to understand the remaining features. Okay. Sex-linked inheritance. Then again broadly of X-linked inheritance and Y-linked inheritance. X-linked again of X-linked recessive and X-linked dominant. X-linked dominant. X-linked recessive, generally mutations are associated with that, say color blindness, hemophilia, Duchenne muscular dystrophy, X-linked dominant disorder, say follicular hyperkeratosis, otherwise called phrynoderma keratosis, otherwise called phrynoderma. Then incontinentia pigmenti, Plus Y-linked are simply holandric characters. Say the sex determining region on Y chromosome, that includes hypertrichosis, porcupine man, etc. These are the broad categories of sex-linked inheritance. I told you already. The characteristic pattern is different in both autosomes and allosomes. We have discussed. So in autosomes, so the probability is same in, that means the probability of inheritance is same in both the sexes. But it is not the case in case of sex, that means allosomes. Depending on the kind of allosome, the pattern may varies. Okay, they are X-linked and Y-linked. The name would imply the only an X chromosome or Y-linked. So depending on the kind of allele, X-linked recessive and X-linked dominant. Recessive allele means with respect to X, female type, I have already discussed. Homozygous condition is required to get expressed. Whereas male, hemizygous, 1 copy is enough. Example, color blindness, hemophilia, Duchenne muscular dystrophy. X-linked dominant is not that much common, that means not familiar or very frequent when compared to the X-linked recessive disorders. Only few characters, only few are known, well known to us, like follicular hyperkeratosis or phynoderma and incontinentia pigmenta. Here, unlike X-linked recessive, here both probability, here both the male and female, one copy is required because dominant, the name it implied. Say if a female get one copy, then the expression, that means the phenotype gets expressed. That means the trait will get expressed. But the thing is that depending on the here, the intensity of dangerous or lethality is different. Males are more vulnerable to lethality when compared to females, because in females there are two X chromosomes now. One normal X chromosome may useful for balance, whereas male just one X chromosome present, so the lethality is high. Respect X-linked dominant. Whereas Y-linked the same, the son get his Y chromosome from father, and common example say the XY gene, sex determinant region on Y chromosome present on Y chromosome, responsible for male characteristics. Say the feature Y-linked characters, what we call holandric characters, whole andric, completely X or absolutely belongs to male. Say like hypertrichosis, hair on the rim of the pin, then porcupine man, straight hair on the body, etc. So it's about different characteristic features of X-linked inheritance. We'll discuss first in depth about X-linked recessive. These are not common, but these are very important because these are very frequent. Okay, X-linked recessive disorders. X-linked recessive disorders. Here we can take one example, color blindness. If you understand one particular example, it is easy to remember, understand others as well. Color blindness. Our retina of the eye contains certain cells which are sensitive to different colors like red, green. Yeah, red color blindness leads to protonopia. Green color blindness leads to deuteranopia. Blue color blindness leads to tetranopia. So here, color blindness is one of the most common X-linked recessive disorder. Here, particularly common in male. I told you why. One copy is enough because of hemizygous condition. Say we have to discuss in a particular case study for better understanding how the inheritance pattern will affect the offsprings and how the skip generation of inheritance is on board or criss-cross inheritance. Say one particular example. A normal female. Capital C is for normal, okay, just like red colored blind, red eyed, same concept. The male are color blind. Small c for color blind, okay. Normal female, color blind, male. Okay. Then the gametes, we got it. This is one kind of gamete, we got it. Same the pattern. We got F1 generation. The first one, that means the female is a carrier and the male is normal. Carrier. The male is normal. This is F1. Then in case of F2, that means if a carrier female when marries with normal male, the condition you have to see. It's called F2 generation. The same as usual, Punnett square. The first one is normal female, the normal male, the carrier female, but affected male. That means 50% of male are color blind. Simple concept, I told you now. Criss-cross inheritance. Criss-cross. Here, he is a grandfather. And he is a grandson. This guy is affected. Grandson. You know, the same concept. This is F2. That means inheritance of a colorblind trait from grandfather to grandson through carrier female. That means through F1 carrier female is called crisscross inheritance or skip generation. I have already discussed. In case of first generation males are normal. First generation males are normal. That means this generation has been skipped off. They are saved. Skip generation of inheritance or crisscross inheritance. Because the ultimate, of course, generally the problem, just like assets, here the disorder has been inherited from grandfather. That means a male parent has inherited his affected gene to his grandson through F1 carrier female. That means his daughter. This is called skip generation of inheritance or crisscross inheritance. And this is one of the case study. The same concept, that means with respect to hemophilia and Duchenne muscular dystrophy also. Hemophilia, what exactly hemophilia? Hemophilia is also a X-linked recessive disorder. We already discussed. There are two possibilities, hemophilia A and hemophilia B. Hemophilia A and B. A is because of deficit of blood clotting factor 8. B is because of blood clotting factor 9. Blood clotting factor. We knew that there are during blood clotting, different blood clotting factors associated in a coordinated manner to ultimately form a plaque, what we call, so that it may cease off the loss. That means formation of plaque, so that there is no further loss of blood. The same concept here, just like the blood clotting factors will be numbered like 1, 2, 3, 4, 5, is like that. Here, factor 8 is essential also, thus factor 9 also. Here, when the factor 8 deficit is there because of this X-linked disorder, then it is called hemophilia A. 80% are hemophilia A only. Whereas if the deficit of factor 9, that means blood clotting factor 9, it is called hemophilia B. So depending on the kind of blood clotting factor deficit, you can categorize hemophilia A and B. The inheritance is the same concept. We need to say this is normal and this is affected. The rest is same. That means because all these are X-linked recessive disorders. The pattern is same. Males are hemizygous, females are homo or heterozygous. The females are heterozygous. They carry. That means they are ultimate. They carry carriers that leads to production. That means they leads to affected male. So the point is same with respect to XY linked. That means sex-linked inheritance. Here, that means the X-linked recessive disorder. Males are much affected. An affected male always shall get, that means inherited his disorder from affected mother. That means affected mother leads to affected male. That is completely, absolute possible. But with respect to female, it is not like that. At least they may save, like becoming a carrier. At least up to one generation. But they are dangerous. But for males, it's not like that. There is no other way out. Because of hemizygous condition, if the mother is affected, the son shall get affected gene. That means affected male is because of affected mother. That's the higher probability. So it's about the color blindness. Then another kind of pattern is X-linked dominant. We are discussed. X-linked dominant disorders. X-linked dominant. X-linked dominant inheritance. Dominant inheritance. Here, for example, I told you already. Dominant, the name it implied, the trait is dominant, the allele is dominant, so one copy is required. But only thing is, in case of male, the vulnerability of lethality, that means the intensity of lethality is high because only one copy. But in case of females, though they also get affected, but since there is another X chromosome which is normal, so there is a genetic balance. So because just like mutuality or dilution, but males are completely vulnerable. But these X-linked dominant disorders are not as common as X-linked recessive disorders. We have already discussed follicular hyperkeratosis, incontinentia pigmenti. Follicular hyperkeratosis is associated with a skin disorder. Here, excess accumulation of keratin that leads to formation of rough cone-shaped structures. Whereas incontinentia pigmenti, it is a random loss of melanin that leads to mosaic appearance of skin. So how it ends with that? Here, the thing is, affected daughter shall always have an affected father. In this case, son is saved because we have discussed Y chromosome always inherited from father to son. Here, imagine father is affected. That means X, some affected gene, Y. The son is saved, but affected daughter become affected. This is very important. And if the mother is homozygous, if the mother is homozygous dominant, for example, and all children are affected, because this will get inherited to all the children. But this is very important. That means if the father is affected, the son will get saved, but daughters are affected with respect to X-linked dominant disorder. Say for example, incontinentia pigmenti.